Namaste all of you. My name is Dr. Ritu Singh Rajput, Faculty of Agriculture and Veterinary Science in Department of Food and Biotechnology, JAYOTI VIDYAPEETH Women's University. In our previous lecture, we had discussed about the stem cells, different types of stem cells and application of stem cells. Now, in this lecture, we will discuss about the recombinant therapy and erythroproteins. Generally, what are the recombinant therapy? In recombinant therapy, therapeutic proteins are the basis of the recombinant therapy. The therapeutic proteins are generally used for the treatment for the abnormal health conditions, and it is replacing by the chemical synthesized drugs, and it has a highly specific functions and less interference and with the normal biological processes, and it is the faster clinical development and FDA approval time, and this is the lessly likely to elicit immune responses. So the base of this recombinant therapy, generally they are provide proteins which is replace the particular drugs. This is also helpful in clinical conditions and treatment of particular cells. So now we told you about the history of the recombinant therapy. In 1918 to 82, the human insulin created using recombinant DNA technology. After that, in 1986, interferon alpha and the muromoneba CD3 approved. After that, 1993, CBER Office Therapeutic Research and the Review form. After that, 1997, the first whole ceramic antibody and first humanized antibody and approved through the authorities. After that, 2002, the 30 billions of share biotechnological drug of the uh, biotechnological drugs are the invented. After that, in 2006, an inhaled form of the insulin uh, uh, um, provide into the market, and have been research on this particular insulins. So insulin also a particular in help in a treatment. Now I will told you about the classification of protein therapeutics. Generally, they have three to four types of groups. In group 1, protein therapeutics within enzyme and regulate. It has in a group 1, protein therapeutics with the enzyme and regulatory activity. In a group 2, it has the protein therapeutic with the spatial targeting activity. In group 3, it will become in a protein vaccination. In group 4 is the protein diagnostic. So this is, first of all, you have to know about the enzymatic activity in group 1. After that, in group 2, you have to know about the spatial targeting sequence. After that, you have the make protein vaccines. Then you have to diagnose the particular proteins from this diagnosis. So now I will told you about the process of the drug production, how to we will provide the drug, and how to we make the recombinant protein which will be replace the drug into the market. First of all, you have to take a cell and a plasmid and ligate, and after that the transfection, you have to culture the cell lines. Then this cell culture mixed with the drug substances which is a crude transfer for the purification. Then after that, the drug purification provide which is the after the purification, and there are so many purification method. It will be, you can be done by the chromatography method, and you can be done there are so many chromatography, ion exchange chromatography, and that methods are based on the particular drug which is related to the particular drug. And after that, the purification, the drug substances which is the properly pure transferred into the formulation for the filling. Then drug product in a sterilization. So just because the particular drug is purely sterilized, the cell line manufacture into the bioreactor, which is which is related to the downstream processing, and medium development has been done on the development and the scale purification. And this is all procedure done in the analytical characterization generally. And the procedure we are using, if we want to do the production of particular substances, we have to use in particular bacteria or from the microbes production for the particular protein production. So basically we can be use the bacillus species of the bacteria, we can be use the actinomycetes species of bacteria, we can be use E. coli, we can be use yeast for the increase the yield of the production, because the bacteria cultures uh, because the bacteria culture helps to increase the yield of the production, it helps to degrade the cells. So this is all about the recombinant therapy, how to we can be provide the cells, how can we make the recombinant therapy, how can we make recombinant drugs, how can we make the recombinant protein. Now I will tell you about the erythroproteins. What are the erythroproteins? This is also part of the uh, therapy. Erythroproteins, which is also called as EPO, is a hormone producing by the kidney that promotes from the formulation and from the red blood cells and by the bone marrow. And the kidney cells can be made erythroprotein, and they are sensitive low oxygen levels into the blood and travel through the kidney, and these cells make the release the erythroproteins when the oxygen level is too low. The low oxygen level may be indicate the diminished number of the red blood cells, and that this cause anemia. Hemoglobin molecules and carry the oxygen through the body. So if in our cells the blood cells in number of blood cell is low, erythroproteins will be make very low in the in the low quantity. So erythroprotein is so much important hormone to produce by the kidney in the bone marrow. Erythroprotein is generally hormone produced by the kidney, as I have told you, and the erythroproteins promote the formulation of the red blood cells in the bone marrow, and the erythroproteins hormones level can be detected and measured by the blood cells. We can be test. So if you want to know about the how much erythroprotein in our cells, you can be done by the EPO test, that is called in a EPO test. And the measurement of the blood erythroprotein level can be used to the detect by the medical conditions. The erythroprotein can be synthesized and used for the treatment of some forms of the anemia. If we have caused anemia, that's why that means you have a less blood cells in our body. So you have to increase the red blood cells. So erythroprotein highly produced by the kidney cells. So erythroprotein has been misused also in so many things by the athletes. So this is the production of erythroprotein is very less, just because this is. And generally the normal erythroprotein range of the 4 to 24 mu ml produced by the cells. And so this is all about the erythroproteins, and I have told you about the recombinant gene therapy. In recombinant gene therapy I have told you about the different the types of therapy, uh, which bacteria we are using in recombinant therapy, which is bacillus, E. coli, actinomycetes and yeast for the production of increase the production. And how we develop the recombinant therapy, how four types of groups of recombinant therapy, which is group 1, where we have to know about the activity of the therapeutic and the regulate. We have to know about the regulating enzyme which is produce the recombinant drugs. After that we have to know about the protein therapeutics, which is target the particular cells and the particular activity. After that, we have to know about the protein vaccines, which is produced by the group third, and after that we have to diagnose the particular proteins. So in this things, we have to know about these things, and these are the classification of protein therapies. And I have told you about the procedure, about the recombinant therapy. First of all, you have to transfer the cell with the plasmid, with the cell lines. After the transfer to the drug membranes with the drug substances, and the after that the purification with the so many methods. There are the electrophoresis method, there the crystallization method. You have to make crystal, we have to use nanoparticle, we have to use many you can use in electrophoresis. It will be gel electrophoresis, it will be SDS method, and you can be use a bioreactor for the production. For the purification, you can be use chromatography, which will be ion exchange chromatography, affinity chromatography. So many chromatography are present, but it's based on the particular drug. It is related to the particular protein, and it is based on the macro and micromolecule. It is present. If it is a macromolecule, it depends on the chromatography. If you use it is if you are using the macromolecules, it is also based on the protein. It will be size, it will be affinity, if it will be depends on its affinity, if it will depends on cations and anions. So this purification method bases on the size, shapes, and filtration. You can also use the filtration method for the purification. It will be ultrafiltration, it will be microfiltration. There are so many methods for the purification. And for the bioreactor, you can be done, it will be airlift bioreactors, it will be so many reactors. For a particular yields, there are so many bacteria. For the culture, it will be, you can also in a production you can be use some nitrogenous factors, some carbon factors. And in a carbons, you have to be set a particular pH for the productions. And you can be set, you can be use biochelators for the particular production. You can be use nitrogen factors, and carbon sources, nitrogen sources is also, and particular bacteria for the production. So these are the basis of the drug productions. And if you want to increase the scale up, you have to use chelators. So many things are used the scale up the particular bacteria. You can be use particular species for bacteria species. If you want to use, if E. coli have been done for produce so many E. coli produce this bacteria in a high level, so you can be use that particular species for increase this yield of that drug production. In a first of all, in a drug production, are insulins introduced into the market as a drug provide, and it is also a protein. And now this is all about the recombinant therapy. In recombinant therapy, they will ask you the normal question, which is what is a recombinant therapy, and the classes of the recombinant therapy. First of all, which protein or which drug be introduced into the market. And in examination, they will be asked the in a large question, explain the process of the drug production. And they will be asked the explain the bacteria, which bacteria is useful in a production, which sources are using in a productions. And they will be they can be asked which sources they can be used, what is the pH. uh, just because if we are talking about the production of particular drugs, particular protein, particular amino acid, particular antibiotics, we are talking about that. So all bacteria are different, all nitrogen and carbon sources are different, on different pH matters, and different kinds of procedure are applied, different kinds of filtration are applied. So you know about this. And so they can be asked anything. And next, we are discuss about the uh, erythroprotein after that. And I have told you about what is the erythroprotein. erythroprotein produced by the kidney, and it helps to increase the level of the RBC, and it can be used as a protein. And if you know about in your cells, the protein cells are less. So in your if you know about the hemoglobin molecules, it can be carry the oxygen from the hemoglobin. And if you know the red blood cells less in your body, that can be causes the anemia. And so you have to treat it. If you want to know how to know about the, you can count the hemocytometer, how much blood cells in your how much blood cells in your, and you know about the red blood cells time duration, it is a 120 days. And you have to know about the as per the questions method, you have to know about what is erythroproteins, and it can be the how to formation of erythroprotein, and uh, uh, what they are responsible for which cells, production of which cells, and produced by the which cells. They can be asked in optional questions. And how to produce erythroprotein, it is also a important questions. And how erythroprotein promote which cells, this can be also asked in a questions. And which tests have been done for the isolation of the erythroprotein. If you know about, you have less quantity of particular cells in your body. Red, if you know about want to know about the red blood cells in your red blood cells, less quantity in your cells, you can test by the EPO test. So it can be asked in a which test will be the diagnostic for the red blood cells. So this is all about the recombinant therapy and erythroproteins. Thank you.